Hi, I'm Richard Sever from Cold Spring Harbor Laboratory Press. With me I have Rick Young from the Whitehead Institute and MIT. Rick, welcome to Cold Spring Harbor.
Thank you very much.
Now, you spoke yesterday about gene expression in regulation of embryonic stem cells and development. And a key component of your talk was this idea of a super enhancer. Now, I think everyone watching will know what an enhancer is, a sort of region of DNA upstream or downstream of a gene that controls its activity. So what's a super enhancer?
Well, in brief, a super enhancer is a large enhancer. It's a cluster of enhancers that seem to work together to some extent to control the expression of a target gene. And we ran into these because we were interested in the transcriptional control of cell identity. And we noticed these very large enhancer clusters associated with genes that many investigators knew were important for embryonic stem cell identity. And that gave us the clue that they might be functionally interesting. And that led us to this view that they play special roles in the control of gene expression.
So it's really a quantitative difference rather than a qualitative difference when you compare them with your regular enhancers. Is that safe to say?
I think part of it's a quantitative difference, but more interestingly to me, it looks like they are aggregating the transcriptional components downstream of the major developmental pathways. So if cells need to sense their environment, and they always do, the signaling pathways come right to these super enhancers, which are then regulating the genes that play prominent roles in that cell's identity.
Right, so they're kind of like a sort of they're higher up the pyramid, so to speak, in the control of cell fate, and a super enhancer is essentially where the whole notion of combinatorial regulation is happening. That's how you get the specificity. Is that the case?
It seems to be. It's also the case that in every cell type we've looked at, the genes that are playing the most prominent roles in cell identity seem to have evolved this clustered enhancer structure. So the sequences are there for the dominant transcription factors in that cell type. And then they seem to be the recruiters of some of the signaling transcription factors that are the receivers for the signals coming from outside the cell.
So these they're distinct from what one would have in the past called a locus control region?
I think they encompass what we have called a locus control region because the locus control regions are also clusters of enhancers. And there are other types of clusters of enhancers that have been noticed before. The difference between the locus control regions and what I think of as the larger group of super enhancers is that the LCRs classically have regulated downstream target genes in different ways, so that during development you might, for example, make the early, more embryonic form of globin during one stage of erythrocyte development, and then the adult globin during another stage. And the different LCRs were capable of differentially regulating those developmentally important genes.
So if you, at the molecular level, are there particular proteins that, beyond kind of obviously the transcription factors that are binding, are there additional protein components that define super enhancers? Do you see different histone modifications? Are there any other characteristics?
The two things that really characterize these for us is that they have an unusual density of the transcription apparatus. And so enhancers, a typical enhancer in my view will have transcription factors, usually a large number in mammalian cells. They will recruit various kinds of cofactors like P300, CBP. They'll also recruit chromatin regulators like BRD4 and switch SNF apparatuses, but they ultimately recruit the polymerase and the transcription apparatus. And unlike the picture we had of enhancers many years ago, we now have come to understand that there are sites where transcription can actually occur and where the RNA components can have functions. And the superenhancers just seem to have a much higher density of that. We've come to believe that's because in three-dimensional space, the clusters actually come together and operate at a point, if you will, where they're aggregating transcription apparatus and then giving it off to the target gene of interest.
And these clusters, are they in cis, or do you get in trans, you get kind of like loops coming in from elsewhere?
Well, because superenhancers are like enhancers in that they can loop some distance to a target gene, they share that feature. And so it seems like the requirement we've seen so far is that they really need to be embedded in a larger CTCF co-bound loop in order to constrain their activity to their target gene. So it's usually a single target gene that cluster of enhancers is focused on.
I see. So you mentioned CTCF. So one of the things that I found fascinating about your talk was this idea of, so it's these insulator regions that the CTCF binds. Is that correct?
Yes.
And correct me if I'm wrong, but they do what they're called. They insulate the effects of the superenhancer. Is that correct?
So a subset do. So of all the CTCF bound sites in the genome, about 35% are engaged in forming these loops. And it's that set that looks like they're insulating. So actually loop formation is insulating. The simple presence of the CTCF protein by itself is not always insulating.
I see, right. And the other feature that was interesting from a sort of general cell biological point of view was I saw in your diagram the word cohesin. And
yes.
the cohesin seems to be at those CTCF sites and also at the superenhancer site. Is that correct?
That's correct.
So this is the same cohesin that I would more typically expect to find at mitosis.
Yes. And so cohesin molecules were originally described as being important for sister chromatid cohesion on the metaphase plate during mitosis and during meiosis. And we came to understand that they actually are also recruited by a component at enhancers called NIPBL to help maintain the loop that's formed between an enhancer and its target gene. So now we've come to understand that the enhancer promoter loops are helped stabilize by cohesin molecules. They can form a ring around two nucleosome-occupied DNA strands. And they also migrate to and are associated with CTCF sites. And so wherever you find a CTCF loop, there's a cohesin molecule co-bound.
So they're essentially a structural, architectural element that fulfills a similar role at mitosis and in interphase. Then.
That's right.
Right.
That's
okay.
correct. They play dual roles then.
And so what does happen at mitosis? Are these structures maintained through mitosis? Does cohesin slide off to somewhere else and then come back to where it was before?
So that's a really interesting question. And the simple answer to your question is we know very little about that process. The two things we do know are that these fascinating topological domains that Bing Ren, Job Decker, Edith Hurt, and others have described, we think they're largely made up of these CTCF cohesin co-bound loops and nested inside additional loops of that sort or loops of enhancers going to their promoters. And that structure, because of the condensation that occurs during mitosis, is sort of lost in these maps of topological domains. They seem to dissolve, and one interpretation is actually the DNA is so condensed now that you see interactions at very high frequency all along the DNA. But you come out of mitosis into telophase and then into G1, and all of those domains are reestablished. And since we think the underpinnings of those domains are the CTCF loops and the enhancer promoter loops, they're nicely reestablished, correctly reestablished as you come out of that mitotic state.
Oh, I see, right.
The exact controls of that are just not understood.
So, I mean, on the subject of mitosis, the description you gave was that these superenhancers function in development.
Mm-hmm.
That they're setting
Yes.
cell identity. Do you see them in normal physiology?
Yes, you do.
Yeah.
You do. In every cell type we've examined, there are superenhancers, and the superenhancers are associated with what investigators have already identified as the genes that play most prominent roles in their identity.
Right. So they're for sort of establishment and maintenance of cell identity.
Yes, and because each cell has a different set of genes that are playing those dominant roles in cell identity. Although the structural components, these CTCF type loops that I described to you, tend to be maintained, the new superenhancers and their looping to their target genes are the things that really change dramatically during differentiation.
And so in terms of kind of abnormal physiology, you also indicated that there was a sort of dysregulation or dysfunction
Yes.
of superenhancers in cancer. So what's going on there?
Well, cancers are, in my view, just a very rapidly evolving cell. They're evolving to adapt to the niche that they live in, and whatever types of mutations occur that give them some advantage, some growth advantage in that niche, are things they'll adopt. And so, because they're genetically unstable, one of the kinds of things they do is they will build superenhancers that are not normally in that lineage to drive oncogenes. And every time we've seen the development of an oncogenic activity, a dysregulated gene activity, we've seen evidence that there's superenhancer formation in the vicinity of that oncogene.
And those are those. You said that superenhancers that you wouldn't normally see in that lineage. Are they de novo superenhancers that you've never seen anywhere before, or are they, as often happens in cancer, recapitulating something that you normally see in embryogenesis?
Yeah, that's a great question, and it's both. You will see, because cancer often represents something of an embryonic state of a cell, a dedifferentiated state, you'll often see that the enhancer utilization for a gene that's typically involved in early development, say an early developmental transcription factor, that is normally silenced in the cells, will be reactivated, and the superenhancer that was driving it will be largely the same. But we more frequently see tumor cells taking advantage of genetic instability to either translocate a superenhancer that's normally being utilized elsewhere in the vicinity of an oncogene, or we'll see insertions or deletions, focal amplifications that are creating superenhancers that normal cells never see.
Right. So it's always kind of gratifying to hear of such abnormal behavior in cancer for people who are looking for, Achilles heels.
Yes.
Is there something about a superenhancer that then makes it a viable target for some form of anti-cancer therapy?
Yeah, well, working with Jay Bradner and Nathaniel Gray, that's exactly what we found, that in many of the most aggressive tumor cells, which tend to have the largest, most aberrant superenhancers, it looks like the larger you are, the harder you fall. That inhibitors that hit various components of the transcription apparatus can have a disproportionately adverse effect on oncogenic superenhancers at concentrations of these drugs that seem to have no effect on normal cells.
On regular cells, right.
So enhancers are really collections of transcription factors that have very weak interactions with the DNA, and they live there through many cooperative interactions they have with one another, and their cofactors and cohesin and other types of epigenetic regulators. And it would appear then that when you have these collections of synergistic, weak collaborative interactions, that there's a propensity to fall apart under some limited duress with these transcriptional inhibitors.
Right, and those inhibitors are we're talking about small molecules, are
These
we?
are small
Right.
molecules. These are small molecules that, at least in the cases where we've investigated, either competitive inhibitors of bromodomain proteins or are catalytic inhibitors of the transcriptional kinase CDK7.
Uh-huh. Well, we wish you the best of luck with that. It's been great talking to you, Rick. Thanks
Pleasure
very much.
to talk to you.
Thanks.